All right, well, good afternoon, everyone. I'd like to introduce an incredible and an esteemed guest who has so graciously donated his time to be with us here for today's seminar, Dr. Ray Wheeler. I met Ray as when I worked as an intern at NASA Kennedy Space Center on space crop production in 2019. In 2020, we both regularly attended a virtual coffee hour where we would engage in these awesome and fun, thought-provoking discussions about the science behind the past, present, and future of crewed spaceflight. For example, one of the questions I had was, would Mark Watney have actually been able to grow potatoes on Mars in that 2015 movie, The Martian? Ray began his career at NASA in 1988. His work in the Space Life Sciences Lab focuses on lighting, the effect of carbon dioxide concentrations on plant growth, and the use of hydroponic techniques for production systems. He's the author and co-author of more than 200 scientific papers and is the recipient of NASA's Exceptional Scientific Achievement Medal. Today, he'll talk about his work on bioregenerative life support systems and their role in the crewed exploration of our solar system. And with that, I give you my former colleague, mentor, and friend, Dr. Ray Wheeler.
Well, thank you, Joseph, for such a kind introduction and the opportunity to participate in the seminar series. So what I will do now is turn off my video first and share my screen. If I can get it. Sorry, kind of struggling here to get the screen up. That's not it. A few technical challenges here. I apologize, folks. Okay, there. Now, hopefully you can see that. Oops.
Looks good.
Good, thank you. I apologize for that. Okay, I thought I'd talk today about some of NASA's interests in bioregenerative life support, and by that I mean the use of biological approaches for providing life support when you go into space and go farther out into space exploration missions. And hopefully some of this will become a little more obvious as I go into this. If you think about keeping people alive in space, it's obviously very serious work. And you have to think about what do they need in terms of inputs. And so obvious things come to mind like oxygen, food, and water. And that water can be both for drinking or even hygiene water. Right now in the ISS, they don't really use much in terms of hygiene water. On the other hand, we're also producing things, waste products like carbon dioxide. It's a metabolic product. They're solid waste we produce and lots of wastewater, things like urine and depending on the capabilities, gray water. And so you can kind of put masses on these. We know some of them pretty well. It depends on body weight and things like that. So it might be something like 31 kilograms per day per person in inputs and a similar number in terms of outputs. So it becomes obvious that that you can't just do this by stowage and resupply on a continuous basis. It just becomes prohibitively costly in terms of your launch mass and just maintaining the supply lines. So a better approach is to use regenerative technologies, and I'll explain a little bit more about that. If you look at the current layout of the International Space Station, and I don't expect you to see all this, but all those colored icons there are components that are involved with life support that are spread around the International Space Station. And right now, all of these systems are based on physico-chemical technologies. In other words, the carbon dioxide is removed with molecular sieves, and then it's reduced chemically through a Sabatier process. Oxygen's generated with electrolysis. And then there's a lot of stowage and resupply. But the point here is that there really aren't any biological technologies being used for life support at present in the space station. And a lot of that's sort of driven by the supply line economics. The space station's pretty close to Earth. We can get things back and forth reasonably easily. But as you go farther out into space and stay longer, those supply lines shift, and your trade economics also shift. And at that point, biological approaches become more favorable. And this diagram kind of shows how you can use, in this case, plants, or it could be other photosynthetic organisms, for biological life support. If you look at the sort of the metabolic needs for humans on that top circle, we need food and oxygen. That's how we get our metabolic energy, and that generates things like carbon dioxide and water vapor as waste products. And then we also need clean water for drinking. Plants, on the other hand, can drive those reactions in the opposite direction. They can use the CO2 from the atmosphere, and then if they have access to water through photosynthesis, they can fix that CO2 into biomass, and then you get you get oxygen as a waste product. The oxygen comes from splitting water that goes into that reaction. The other thing that might not be immediately obvious is that plants are always transpiring water. So they're pretty good stills, actually. And if you're clever in how you approach this, you might pre-treat some of the wastewater, but then you could use the plants as sort of a finishing step where they generate distilled clean humidity that can be condensed out as clean water. So these concepts aren't new. The novelist Percy Gregg wrote about using plants in the late 1880s, 1800s. Tsiolkovsky, a famous Russian space scientist and philosopher, wrote about it early. And then if you go all the way to the bottom of that list, a fellow named Jack Myers at the University of Texas really began to kind of push this in a more serious way with studies with chlorella algae. In other words, can you use photosynthetic organisms, in that case algae, but also plants, as a potential a tool for life support in space? I'll go back even a little bit further to the 1700s, just with some quotes from Joseph Priestley, who sort of co-discovered oxygen. He didn't specifically identify it, but his work kind of revealed its presence. And I'll just read through some of these quotes. "I've been so happy as by accident to hit upon a method of restoring air, which has been injured by the burning of candles. And I've discovered at least one of the restoratives. It is vegetation. When I first put a sprig of mint into a glass jar standing inverted in a vessel of water, it continued growing for some months, and I found that the air would neither extinguish a candle, nor was it at all inconvenient to a mouse." So Priestley put a mouse under a bell jar with a plant, and the mouse was able to hang in there longer. So we've kind of called him our patron saint of bioregenerative life support systems. The interest in these kinds of approaches is not just limited to NASA. It's been a worldwide interest through different space agencies. You see a timeline here, and you can kind of see that the US and the USSR, and then later Russia, of course, have been some of the longer standing countries involved with this. Europe, Japan, Canadian groups, And more recently, the very bottom arrow there shows the Chinese National Space Agency is very actively looking into bioregenerative life support systems. Just out of curiosity, if you're interested, NASA's efforts really ramped up around 1980 in terms of plant research for bioregenerative life support. And you can see some of the universities that were involved, Utah State, Tuskegee, Wisconsin, North Carolina State, UC Davis, Purdue, and others. And then some of the NASA field centers were also involved. Just, you can, this will be recorded, so if you want to go back and look at this later. Okay, plants for space, crops in particular for space. If you're going to use plants to generate oxygen and remove CO2, you might as well choose crops. You want your biomass to be edible, so you want it to be multifunctional. What are the attributes you want? Well, you want high yielding and nutritious plants. And there was a lot of focus early on macronutrients like carbohydrate, protein, and fat, but more recently, we're also interested, at least for starting missions where we're only going to grow a few supplemental plants, can we target secondary metabolites like antioxidants, lutein and zeaxanthin, which are important for eye health, and some vitamins that might be lacking or have a limited shelf life in space food systems. You always want plants with a high harvest index. That's a term referring to the edible to total biomass. So leafy greens like lettuce are largely all edible, 90% of it maybe, but they're not all that nutritious. Seed crops like wheat and rice, things like that, you're doing pretty well if you get maybe 40 to 50% of the biomass in seed. So the higher you can keep that harvest index, the better. In space, we're limited in terms of volume and vertical dimensions, so we're interested in low growing or dwarf crops. We have to think about the environmental considerations for the crops that we are interested in, the horticultural considerations, planting, watering, do they require pollination? How do you propagate them? And then are there a lot of processing requirements involved with them? So these kinds of considerations. Again, this is going to be a hard thing to read, but a point I just want to make here is lots of groups have thought about the types of crops that might be useful for space. NASA's done it, Russia's done it, the Japanese, European space agencies and others. And so there's a mix of sort of, you might call staple or agronomic crops, and then some supplemental food crops for fresh vegetables, things like that. It's interesting though, you can see cultural differences. For example, that fourth column, the Japanese, they're fond of vegetables like shungiku and komatsuna, rice and soybean as well. And so it's kind of a fun thing to think about as you have multicultural crews that go into space, how you might compose a diet and the plants that would provide some of that. NASA has sponsored some crop breeding work. Some of this was done at Utah State University. So these pictures are courtesy of Bruce Bugby at Utah State. A lot of the selections of varieties have just come from existing genotypes. So you can see an example on the left there of just the genetic variation in rice, for example. You see that ultra dwarf rice, turns out it doesn't yield very well, but the potential is there for very short varieties. And then the group at Utah State did some breeding work with wheat. And you can see that their final product, perigee wheat, is very short and has a high harvest index. It's only about 40 centimeters tall. So that's good. That's what we want for space. In addition to conventional breeding, there are lots of molecular biology tools now, genetic engineering, gene editing. And I'll just show one example here of some collaborative work we've done with the USDA. We never really thought much about fruit crops, tree crops, because they take too long to get into production. They'd be too big. Through some genetic engineering work, a group at USDA's Kearneysville station in West Virginia overexpressed the flowering locust complex gene in plums. They wanted to go through their life cycle faster to speed up breeding. But what this ended up producing were plums that would go to seed to flower to fruit in something like 12 to 18 months. You can see that pot there in the middle. That's a young plum tree that's already begun to flower, and they didn't have any dormancy requirements. So it just shows that, you know, you can use a lot of your different tools to even include crops that you might not normally consider for space. One of the things we have to think about in space settings is we have to maximize and recycle all of our water, our air, our nutrients. And so a lot of what NASA has sponsored in terms of crop research has used recirculating hydroponics. This allows you to use the water continuously. You don't waste any of the nutrients or the fertilizer in that. And so, and this is widely practiced commercially, but it just fits what we have to do in space. You certainly can't be wasteful of the water and the nutrients that you bring along. And so we tested this with a lot of agronomic crops, things like wheat, soybeans, rice, sweet potatoes, and it worked very well. It even worked for root zone crops like potatoes, you see here, peanuts. In this case, we used a form of hydroponics called nutrient film technique. So you only have a thin layer of water, a nutrient solution running through the trays. Potatoes don't like to be too wet, and this worked great for potatoes. So, hydroponics isn't new, but using them for these types of crops was kind of novel and hadn't been tested very much. If you think about weightless settings, so like on the space station or when you're traveling somewhere to get to Mars or to get to the moon, you won't have gravity, so you won't have normal drainage of water. There's no up, there's no down. And so you have to think about, how do you deliver water to the roots of plants? There have been different methods that have been tried. One of the ones that seems to work pretty well is you use porous tubes that you can use to contain the water, and then you can allow the plant roots to either wrap directly around those porous tubes, almost like a chia pet, if you're familiar with those things they advertise at Christmas. Or you could use the tubes that are embedded in a soil, a medium that you could use then to irrigate that medium. But in both cases, you're containing the water, so you don't have any free water that can float around and escape. It makes it more difficult and more complex to water the plants, and this is still an ongoing challenge for weightless settings. But just to show you, when we've used hydroponic techniques to track water, you get these kinds of data, time on the x-axis there, and then the amount of water used. And it's quantified as liters per square meter per day. In this case, potatoes, and two experiments where we grew them at different light intensities. But you'll notice as the stand fills in, they use more water as the stand grows higher and gets closer to the lights. But look at the enormous amounts of water that go through plant stands. This is like 10 liters per square meter per day at its peak. So there's a lot of water use, but it's not like this water is just going away. You can condense it and then either recycle it right back to the plants, or you can send the clean condensed water to the humans, retrieve some wastewater and so forth. But point being, there's a lot of water vapor moving through plant chambers. If you compare it to, say, CO2 or oxygen in terms of photosynthesis, there's probably 2 orders of magnitude more water vapor. Water is the #1 gas moving through plant chambers. As I mentioned, we can even couple the plant systems in terms of treating wastewater. The upper left photo shows where we grew some plants and we added gray water that we had a surfactant soap in it. And we even, I say we, the microbiologists in our group deliberately added some human-associated organisms to see if they would persist in the system, like in the root zones of the plants. And so we could do that. The plants could tolerate things like gray water. The bacteria in the root zone could break down these surfactants. And it turns out the human-associated organisms didn't compete very well in these kinds of environments and in these plant microbiome communities. It's just not where they naturally occur. And so a lot of them typically just drop below detection limits. We can use biology in other forms, like in bioreactors, to directly treat wastewater, things like urine, for example. And a picture in the bottom right just shows some studies that we carried out. And this is still an active area of research for NASA. And honestly, I think bioreactors are probably one of the most competitive technologies for early use in space settings. The challenge with bioreactors for wastewater, and it's like a municipal water treatment system, you have to start them up and you have to build up the microbial communities. And once you're up and running, that's good, but they're not conducive to kind of turning them off and going dormant and turning them back on. And so we're having to kind of figure out how you would do that if you were forced to go into dormant stages for periods. So now let me show you a specialized chamber that we had at Kennedy Space Center. This was built in the mid to late 1980s, and then it ran for about 12 years to about 2000. So a lot of this is dated in terms of the photos and the information, but I think it's very relevant. The chamber was tightly closed atmospherically, and that's one thing we needed in terms of understanding how the plants would perform in closed systems like space. We used recirculating hydroponics. You see the tanks there in the lower right. And then you can see some of the information on the chamber there. It provided 20 square meters of plant growing area. So it was a pretty large plant growth chamber. And if you look inside of this, the way it was arranged is there were 4 shelves where we had hydroponic trays, and then there was a light bank above each shelf, and then another shelf of trays and a light bank. And then you can't see the upper chamber in this photo, but 2 more. And at that time, we didn't know it. The term didn't even exist, but this was essentially a form of vertical farming. Maybe you've heard that term used now. This is an expanding area of controlled environment agriculture that's conducive to urban settings where you don't need a big footprint in terms of area, but you can be very volume efficient in terms of how many plants you can produce. And so that's exactly what we were hoping to do in the NASA studies, was have a volume efficient approach. In other words, not only quantifying yields per unit area, but also per unit volume. And so to my knowledge, this is probably one of the first working examples of a vertical farm. And it operated for 12 years. It wasn't a commercial effort, clearly. It was a research effort. But something that who would have thought NASA might be out in front of, something that's quite popular nowadays. We tested a range of crops like wheat in there. You can see there's no soil in these systems. There's mature wheat in this bottom right photo. Soybeans, I showed that one photo earlier. Here I opened the door and you can see down, you can almost see all 4 levels of the trays, the hydroponic trays. Lettuce is a classic hydroponic controlled environment crop. It did very well in the system. I showed you earlier, we could even grow potatoes in this and root zone crops. And they did very well. I'd had previous research experience at the University of Wisconsin with potatoes, and I didn't think it would work, but this nutrient film form of hydroponics was great for potatoes. And at the bottom there, you see after we cut off the stems and we just roll back the plastic cover to these trays, the potatoes were just sitting there and with no soil, nice clean ways to grow the tubers. And it turns out that this has caught on with seed potato growers. Seed potato growers it's sort of a parallel industry with potato production growers. The seed potatoes are the propagation stock that are used for field growers. And the seed potato industry likes to either be in very remote settings away from production fields, or to do this in clean, controlled environments because they don't want to get virus and fungus building up in their propagation stock. Potatoes are all clonally propagated. And so now this hydroponic approach is being used by several, that I know of, at least groups in the US and also around the world. So again, a sort of a technical spin-off, you might say, from NASA. So, okay, we've seen the crop growth. What are the plants doing in terms of their oxygen production and CO2 removal? So here was a typical set of data. Time on the x-axis there, and you can see the light and dark cycles. It's 12 hours light, 12 hours dark. These were soybeans. And on one y-axis is oxygen production. So you see in the light, the oxygen goes up, and so that's from photosynthesis. In the dark, it goes down a little bit because the plants are always respiring, just like any organisms. In the light, it's sort of the photosynthesis overwhelms that, and so the net effect is the oxygen goes up, but it comes down a little at night, then up in the light and down and so forth. And by the way, if you keep the chamber closed long enough, the oxygen gets to levels that become a fire hazard. So we either had to scrub the oxygen out or just open the door to vent it. But they were doing their thing. CO2, on the other hand, would build up at night from respiration. And then when the lights came on in the morning, you could see it comes down very quickly. I don't know if you can see my cursor. It comes down very quickly in the light. And then we began to add CO2 to hold, in this case, 1,000 parts per million. So we wanted to control the set point so it wouldn't get any lower than 1,000. Because that's a good concentration to promote and very high photosynthetic rates. So they were doing just what we expected them to do. You could track this throughout the growth and development of different crops, time on the y-axis, x-axis here, and then CO2 exchange rates on the y-axis. And so positive values in this case indicate net photosynthetic fixation of the CO2. And then the negative values are the respiration rates that occur at night. And so you see two different light intensities. I think these are soybeans. The photosynthesis goes up as the stand fills in and they get up close to the light bank. And here, this just shows what happens in terms of horticulture. The first time we tried it, the stand collapsed on us. It lodged. So the whole stand flopped over. And so we had a dip in our photosynthetic rate because the stems and leaves weren't oriented very efficiently to intercept the light. They kind of fixed themselves and then kept going. So these kinds of gas exchange curves are very informative in terms of what's going on in the chamber. Are there perturbations? They're very good indicators for stress and events of that sort. So we did a number of studies with different crops. You see wheat, soybeans, potato, lettuce, tomato. We did some rice and radish. I didn't plot those. And then plotted here on the x-axis is light, photosynthetically active radiation, and then a yield rate on the y-axis, so grams per meter square per day. So that's grams of dry mass. And it's pretty clear here that light has a very strong effect. It's essentially linear in this range. The noise in the data here are largely due to the different crop species, but you see the more light you give it, the more yield, the more biomass you get. And then a portion of that biomass is edible. Again, that depends on the types of crops. And just to kind of give you a calibration point, a bright sunny day on Earth is about 60 moles per meter square per day. And then Mars is about 28 to 30. So Mars only has about 43% of the light that reaches Earth, but that's pretty good light. It's probably more light than you get in Blacksburg, Virginia in January. So anyway, these kinds of data are very helpful then in terms of planning and anticipating what we get. So light's really important. And that means if you're doing this with electric lighting systems, that's a huge energy cost. And when we started these studies, we were using high pressure sodium lamps. And so a group with NASA funding at the University of Wisconsin began to explore light emitting diodes, LEDs, as a means for growing plants in controlled environments. They wanted to do it in little chambers that they sent up in the shuttle. And this was in 1990, and they patented that concept. And so the LEDs weren't very efficient at that time, and good blue LEDs didn't even exist. But the LED industry has moved ahead so fast, especially in the past 10 to 15 years. The original LEDs that were maybe 5 to 10% efficient now, the reds and blues, they're probably 70 to 80% efficient. So it's just remarkable how much LEDs have improved in terms of their electrical efficiencies. So 70% efficient, if you put 100 watts of light or power into an efficient LED, you're going to get 70 watts of light out. Sodium lamps are maybe 30% efficient. So this was a huge development in terms of reducing the power demands. And again, now LEDs are used around the world for growing plants in controlled environments. So this was a NASA patented and developed concept, and it was researched by NASA quite a bit in the 1990s. It would have happened anyway, but again, NASA was kind of out in front of the curve on this. It turns out, I think the patent that the University of Wisconsin had, I don't know that they ever made a nickel off of anything, because the LEDs just weren't very efficient and they were very expensive until the past 10 or 15 years. But another way you could go in space is maybe if you're in a protected structure, you could collect the light, solar light, externally with things like parabolic mirrors. You see here a demonstration that we had on the roof of a building. These primary mirrors would reflect the light to a secondary dichroic mirror here where my cursor is. That would let the long wave light go through. The visible would then reflect back onto a fiber optic bundle. You see right here. And then these fiber optics would run down into the building underneath, and then we could deliver the light to a plant chamber. And when we had brand new mirrors that weren't dirty or tarnished, we could get 40 to 50% of the incident light. These mirrors would track the sun across the day. And so you can use direct light even. It would depend on the setting where you are in space, of course. We've even looked at things like not just temperature, CO2, and light effects, but something you might have to consider for space is we might not be at 1 atmosphere pressure. And there's a range of reasons for why it's worth considering lower pressures, like 2/3 of an atmosphere, which would be about 67 kilopascals, or maybe even down to 8 psi, which turns out to be 55 kilopascals. At these lower pressures, you can do what are called rapid EVAs. That's essentially a spacewalk. When they get in their spacesuits right now at 1 atmosphere pressure on the International Space Station, they have to do a prolonged pre-breathe, the astronauts, so they don't get the bends when they go out into space. The spacesuits are at a real low pressure, like 4 or 5 psi. And so if you drop the pressure low enough and maintain the oxygen partial pressure, they can essentially suit up and go right out the door in an emergency. Right now, they can't do that without some medical risk. The other thing is it reduces your leakage. You can maybe alleviate some of your structural demands. So back to plants. How do plants respond to reduced pressures? Turns out they do pretty well. We've tested, and many others have tested plant growth at reduced pressures. They can go down to 20% of an atmosphere and do just fine. You see here some plants grown at 1/3 of an atmosphere. provided you keep the oxygen partial pressure above, say, 15 kilopascals. So the equivalent of, instead of 21% at 1 atmosphere pressure, 15%. So the plants, they tend to suffer a bit in terms of root respiration and dark period respiration if they don't have sufficient oxygen. But they're very, very tough and can do quite well at low pressures. In fact, we even exposed them sort of to catastrophic pressure drops. We pumped them down as quickly as we could, and then let them sit at about 1 to 2 hundredths of an atmosphere at that little red diamond right there for 30 minutes. So say we had a space greenhouse and we lost pressure catastrophically. And so the pressure got really low here, and now you just have boiling water that's providing all the atmospheric pressure. If you can maintain the temperature, they survive quite well for 30 minutes when we repressurize things. So this sort of an event would be lethal to humans, but the plants did just fine. So that was kind of a neat thing to know. Oops. Okay, so I've talked about providing the oxygen and food and so forth. But there are other things that might be important in terms of having plants on space missions. Even if you only have a small area, growing area, you could still provide fresh foods. We really don't have good means right now for preserving foods, and we don't have refrigerators on the International Space Station. They're considering some for future missions. So if you could provide on a steady basis just some fresh, crunchy foods that have colors and textures and flavors that you don't get in the preserved packaged foods, this could be really important to human well-being. Just the mere fact that you have bright light and you smell the aromas of plants, you sense a higher humidity around them. And maybe they can even provide a diversion or a recreational activity. So these ancillary effects are pretty important, we think, on long duration missions, but we need more data to see how important they are and how they factor in terms of the different trade studies. And there we go. So here's a good example of it. At the US's Scott Edmondson South Pole Station, there's a plant chamber where they grow fresh foods for the overwintering crew, and they provide that to the galley there on a continuous basis. But they have this anteroom where the crew members that are at the station can go sit and relax. And you see here a husband and wife team that were on this particular overwintering mission, kind of having a dinner a night, and they're sitting in this anteroom where they see the bright light, they sense the humidity. The atmospheric humidity in the Antarctic is low to begin with, and when you bring it inside a protected habitat and warm up the air, it's even lower. And so your sinuses are dried. So the point being here is that there are these other effects, you might say. And E.O. Wilson, the famed biologist at Harvard, has even written about these, the concept called biophilia, where he's saying that as humans, we're intrinsically linked to other living organisms in terms of our well-being and our behaviors. So where are we now? We have small chambers on the space station. One of them shown here, it's called Veggie. It's a very simple chamber. It just has an LED light bank, and then the plants are grown in little soil packets. We call them pillows. Those packets then sit on a wet surface that has a capillary supply of water. Hasn't worked very reliably, but and then we put time-release fertilizer in there. And so this has become a means of providing some fresh vegetables for the astronauts on the International Space Station. You can see the red and blue LEDs there that are being used. So I think if we think about using plants and bioregenerative life support as we go farther out in terms of exploration, it'll be sort of an evolutionary development. We have Veggie now that's only 0.15 square meters. There's a concept called a salad machine or a vegetable production unit and we're looking at a system like this for the International Space Station, where now maybe you scale up to 1 or 2 square meters to provide even more food. Maybe you could attach a whole module that could have a larger dedicated growing area like there in the lower right, and then eventually get to a surface setting where maybe you repurpose a logistics module and use it for plant growth, or you have it part of your habitat or some other structures where you now begin to really expand up in terms of your plant growing capabilities. So a few lessons learned here. We know that if we have high enough light, with 20 to 25 square meters of crops, we can provide all the oxygen for one person. And if you double that area, you can probably provide about all of their food. And by food in this case, I mean their dietary energy, their calories. And that's assuming about 2,500 to 2,800 kilocalories per day. High light, that's key. Remember that light is driving photosynthesis, and the productivity goes up with light. We still need better adapted crops, though. We know that some short growing cultivars have done very well, but we need more of them. We have to keep improving the harvest index and targeting certain nutrients to improve those, and use all the tools we can. Conventional breeding, selecting amongst existing gene pools, molecular techniques. I think we need to continue to work on energy efficiency, especially with lighting, but the improvement with LEDs has just been remarkable there. In any controlled environment agriculture, CEA, sorry about that, CEA, controlled environment agriculture, you need lots of water, at least to prime the system. And this has to be recycled. So we need to understand more about integrating water recycling loops with wastewater management and being able to operate things on a sustained basis. We need fertilizer to grow the plants. A number we've come up with is something like 90 kilograms per person per year to grow their own enough food for one person. So where are you going to get that? You need to maybe prime your system initially, but you'll have other resources. You'll have waste streams. Things like the inedible biomass of the crops you've already grown. Can you recycle nutrients from that? Can you take the urine that the humans produce and recycle nutrients? It's a great source of nitrogen. So we have to get innovative in terms of how we reduce the import needs for fertilizer. Can we use in situ materials, the local regolith or rock? Is that a potential source of nutrients? Maybe. It's kind of crushed rock, so there's not a lot there, but there could be some useful compound and elements. We need more data on the psychological and sociological benefits of plants and what that conveys to humans. If we can avoid the astronauts of going into periods of lower productivity, where they're just kind of get into a funk on a long mission, and they're not working at their optimum, that's a cost in terms of missions. And so if you can keep their productivity elevated through ancillary effects like having plants, that's a genuine benefit. And then I think we have to keep in mind that no one's going to set up a big space farm right when we get to a setting like the moon or Mars. It'll be a sequential development, sort of an evolutionary thing. NASA's had a lot of success stories through their research. A lot of this was largely through funded research to universities. We've shown that we could produce really high yields of crops in controlled environments. You see some numbers there where we could exceed world records with high light, CO2 enrichment, use of hydroponics. Technologies like LEDs and use of recirculating hydroponics have now spun off into applications in commercial terrestrial use of controlled environment agriculture. And so I think as we go farther into space and stay longer, I think you'll see we'll shift more from the current mode of using physico-chemical systems that we have on the International Space Station and use more and more bioregenerative systems. And really, that's the only way we're going to get to achieve higher degrees of autonomy and Earth independence. And so with my last slide, I'd just like to thank some of my colleagues. This is, as I say, this is a dated picture, but these are colleagues that I worked with when that large chamber was largely operational, and the extended university research community. So thank you again so much for this opportunity to speak to you today. And why don't, at that, I will stop sharing. And I will now turn my camera back on. And if people have questions, and if there's time, Joseph, I'd be happy to answer any questions.
Yeah, absolutely. It looks like we've got about 14 minutes for questions. So thank you for that amazing talk. The first question that we have that was posted in the Q&A was, what is your idea on how many different plants will be in the mix that is useful for space, and what kind of additional plant genetic engineering do you think will be useful?
I think we need a broad mix of plants. We can't take everything. I mean, not hundreds of species, but dozens, tens, something like that as you expand up. What'll be different about, say, a space system versus a commercial controlled environment agriculture system, where they focus largely on monocultures, because that's a good way to optimize your whole system and performance. You grow lettuce, or you grow arugula or something like that, or tomatoes. We're going to have to grow a mix of species. They're going to have different light requirements, different light optima, you might say, and different temperature optima. And so can you do that in one common environment, or do you need partitioned environments? These are questions that we still don't know. Can you mix them together in a common nutrient solution? Do you see allelopathic effects, sort of species to species competition or chemical warfare? Again, we don't know. So those are looming challenges. I think initially we want to target supplemental crops that add perishable foods to the diet. As we expand up, then we want to begin to infuse more and more of the macronutrients, the carbohydrate, protein, and fat into the diet, as to really reduce the import needs of food. So genetic engineering, gene editing, I think we need it clearly. There aren't any established guidelines yet for use of GMOs or GE plants for space. I think everybody is clearly in favor of it, to use all the tools possible. We won't be constrained with, say, some of the issues that you might say GM crops on Earth are. We're not worried about the plants escaping or the pollen blowing around. I mean, a space environment's about as contained as you get. But nonetheless, we're going to have to establish some guidelines there, and what's going to be allowed for consumption. But it's being tested already, not in terms of human food, but certainly being tested in terms of improving the crops today. Focus on nutrients for now, but these dwarf growth traits, partitioning more into edible structures, all those attributes that I noted earlier. So that's a great question.
Thanks for that. The next question: have any plant pathogens ever made it as a stowaway to the space station?
Yeah, they have, and this is a great question. By nature, growing plants, and you have humans there, you're going to have mixed ecosystems is at the microbial level. And really they're going to be diverse, and you want them to be diverse, and you want them to be stable. You don't want them kind of drifting. And it's an interesting exercise in island biology when you think about this, because once you kind of close the door and go off to Mars, you won't have any more infusion of genetic material. But are there some bad actors? Are there some pathogens that can get in? The answer is probably yes. And we know for sure that we've seen one pathogen, a fusarium fungus that affected some zinnia plants that we grew in the veggie chamber that I showed you. It was kind of an opportunistic pathogen. And it turned out it really took off when the chamber control failed. The air circulation fan failed. And so the humidity went way up. We were getting condensation on the leaves. And so that made it much more conducive for this fusarium organism to grow and then become pathogenic on the plants. So it's there. You're not going to get rid of it now. I think the key is you want quality control when you start loading up, say, for a Mars or lunar mission, in terms of you want to try to do everything you can to keep known pathogens and aggressive organisms out. Are you going to be completely sterile? No. Do you want to be completely sterile? No. Again, you want, as I said, you want to probably maintain diverse and stable ecosystems. That when I showed you that example of adding human gray water and shower water directly to the plants, the human-associated organisms didn't compete very well in there. And these were, potential pathogens for humans. That was what we were concerned about. So, but plant pathogens are another thing. And so we just have to be very cautious and try to be as careful as we can. That's a great question. Thank you.
Next question. When growing plants hydroponically, how do you start them? Do you start them on agar plates or directly in the hydroponic system? And then how do you keep the algae growth down?
Yeah, okay. You can start them in different ways. We chose to start them directly in the growing system, and we used nylon wicks that would dip down into the water, and we had little covers over the trays to elevate the humidity. It turns out that's not a very efficient way to use your area. You don't want to use your primary production area to start your seedlings. You're far better off in terms of volume and area management by starting your seedlings in a nursery, for example. And so these could be started in little plugs of some material where you don't need a lot of it. Or we could even start them again on wicks where we're not using any solid media, and then you transplant them out to the final spacing, maybe at 10 or 15 days age, and you save that early wasted space. So there's a variety of ways. We haven't started any in agar. What we did with our potatoes, we did propagate those in vitro. We weren't using potatoes that, where we use tubers as the propagules. I think for long duration space missions, though, you would probably use tubers as propagules. You wouldn't want to take a whole tissue culture system with you. I think it'd be extra complexity. And you could just save the little bitty tubers, for example, as propagules. So a variety of ways to do this, but propagation is a big concern, and what are the optimal ways to manage it and get the most out of your available space is what you really want to do. So good question, thank you.
Is radiation an issue with causing mutagenesis, and if so, how do you maintain stable lines of your plants?
Yeah, we don't know. There have been radiation studies with plants, especially in the 1970s and 80s, after nuclear power was really on the increase. There were a lot of studies. And clearly there's interest in human exposure to radiation as we go beyond low Earth orbit and we get beyond the magnetic fields that protect us in Earth. And we're sort of in interplanetary or cislunar space, you're fully exposed to solar events, cosmic rays are omnidirectional. So it's sort of the worst case when you're sort of transiting somewhere, when you get to the surface setting, at least half of your horizon is protected from cosmic rays. And the farther you get away from the sun, you get a little more diminishment in terms of solar particle events. So if the environments are safe for humans in terms of the shielding and whatever measures are taken, chances are they'll be safe for the plants. Can you have separate plant structures with less shielding, where you have less mass around them, or maybe even transparent structures that can hold pressure that don't have much shielding at all? Or could you put water ice over it? People have talked about different ways of providing shielding. So we're concerned about it for plants. We don't know what all the effects are in terms of high exposures to radiation. We think plants are pretty tough. The thing about plants, you don't have to be ethical in some ways. If you lose a few individuals, you might say, that's, if the main population survives or doesn't become mutated, you're still okay. So a lot of unknowns there, though, and that's an active area of research that we need to continue. Yeah, excellent question.
So we might only have time for one or two more questions. The next one would be, what do you consider to be of the remaining challenges to solve, what is the biggest one or the hardest to solve?
Yeah. I mean, there are the upfront challenges of mass, power, volume, and we're working on those. And so it's hard to know exactly which one will be the key one. Honestly, I think demonstrating sustainability is probably in the long term one of our biggest challenges. If you set up a system in space, a space farm or a greenhouse or a plant growth structure, and you operate it, will it still be producing at the same rate, a year later, 2 years later, 5 years later? What are your life cycles in terms of the equipment and the microbiome that builds up in these systems? And those are all unknowns, and these are really long duration experiments that need to be carried out. We need to set something up in a high fidelity way and run it for 5 years or whatever. And understand what are the failures, are the systems sustainable? You might get a different answer from somebody else, but I'd say sustainability.
All right, it looks like we just have a minute left. So last question, how do you handle, how would plant waste be handled in a spaceflight environment?
Yeah, that's a great question. If you scale up to produce a lot of your food and life support with plants, turns out your number one solid waste you're probably going to be producing in that habitat is inedible plant biomass. And so what do you do with that? And we've looked at ways of degrading it in bioreactors, stirred tank reactors, composting systems, using pyrolysis techniques, whatever. What we have found, at least with bioreactors, is we can retrieve a lot of the nutrients that are in that inedible biomass, and then we can recycle that right back to the plants in the nutrient solution. And some plants that have a lot of inedible biomass, we could recycle 50 to 75% of their needed nutrients back. So that's one way. The residual cellulosic lignin type materials, they're pretty tough to break down, but maybe you could get clever and use that in some way. Maybe you could use it as a building material for 3D printing and bind it into some kinds of materials that would help you in your habitat. So I think we need to get innovative and clever about that. The other thing people have talked about is using those leftover biomass and feeding secondary organisms like fish or insects or fungi, and then trying to retrieve some food value back out of that. And so those are other considerations. And the Chinese and Japanese have used insects, for example, as sort of secondary producers when they have inedible biomass. So a great question. These are all great questions. My gosh, I could
Yeah,
keep going here.
I wish we had more time to get through all of them, but I want to be respectful of everyone's time. So, Ray, thank you so much for donating your time to be here with us today. We really appreciate it. This was a lot of fun. Thank you.
Thank you, Joseph, and thank you, everybody. Bye.